will be out as a paper in a few weeks. So, so with that, let's dive into the spatial immune landscape of melanoma. So to start, melanoma. This is a cancer that derives from the melanocytes in the skin, one of the many cell types that make up your skin. An accumulation of genetic defects can cause these melanocytes to turn into melanocytic nevi, colloquially known as moles. And in certain cases, these nevi can undergo melanocytic transformation into melanoma. Now, in this case, an early stage melanoma, stage 1, it's highly treatable. You remove the surrounding tissue and the prognostic is extremely good. However, that's a different story in later stage melanoma, more advanced melanoma, where there is migrating tumor cells and you have metastasis. Now, historically, previously, there was the main treatment for this type of melanoma was targeted therapy, which often initially had good response, but actually developed mutations to be a treatment, and the disease would progress. However, recently, so the just about 13 years ago, types of drugs have come out to treat melanoma, and it's applied to many different types of cancers, called immunotherapy or immune checkpoint inhibitors. So the first of which was ipilimumab, which targets the CTLA-4 checkpoint, and nivolumab, which targets PD-1. And basically the way these drugs work is they target these particular checkpoints in the immune system and activate immune response to the tumor. And as you can see from these survival curves, these drugs work extremely well. This advanced melanoma went from almost a death sentence to a almost curable, or at least in some cases curable, or at least a long-term survival is possible using these drugs. However, there's still, up here, there's still about 50% of people who just don't respond in any way to these drugs, and that's a pretty significant population. So a very active area of research is why do some patients respond well to immunotherapy and why do some not? So an important aspect in response to immunotherapy is the immune microenvironment within the tumor. So is the presence of different types of immune cells within the tumor area. Here I'm showing some example images from a paper published in 2014 in Nature, which found that in treatment with anti-PD-1, certain patients with higher numbers of CD8, PD-1, and PD-1, which is the cognate ligand for PD-1, cells that express these proteins, responded better to anti-PD-1 therapy. And additionally, they found that close proximity between PD-1 and its ligand was also indicative of better response. So you can really see how the presence of particular immune cells and the spatial relationship between them in the tumor can be very important in determining what the response is to these particular drugs. One limitation, however, is looking at this type of data. What I'm showing here is immunofluorescence data. And here we're only able to mark at most 7 proteins. So if we're trying to identify tumor and many different types of immune cells that are present in the tumor, this is very limiting. So that brings me to the technology that I'll be presenting today, called Cytometry time of flight imaging mass cytometry, or CytOF IMC. The way this technology works is, well, first of all, the main benefit is that it allows us to probe for many, many more proteins than we have with any previous technology. As I said, with the highest number that you can have for immunofluorescence is basically 7. And now what I'm showing here is 35. So I'll explain how we're able to do that. So first of all is designing a tissue microarray, which is basically just a slide with many cores from all the different tumors or other types of tissue that you want to image. Then we stain with antibodies, similar to how you would for immunofluorescence, but in this case, rather than having antibodies that are bound to fluorophores, they're conjugated with heavy metal isotopes. Then the slide is put into the machine where a laser will ablate the tumor samples 1 micrometer at a time, 1 micrometer squared. This creates an ionized isotope plume that is then picked up by a mass spectrometer, which can give us the counts for the number of metal isotopes present in each of these spots, which gets translated to a pixel in an image stack. In this image stack, we have each layer of the stack corresponds to 1 marker, and each pixel within the image has an integer number for the number of isotopes present. So this tells us fairly directly how many antibodies were bound in that particular area. So in this project, what we did was we applied CytOF imaging to 72 samples. We used 2 microarrays. The first one was a commercially available tumor microarray. This included 5 nevi samples or moles, 6 acral samples. Acral skin is non-glabrous skin, such as the palms of your hands, the soles of your feet. Also non-acral skin tumors. So this is other types of skin, normal hair-bearing skin. 13 mucosal melanomas and 11 lymph node metastasis for a total of 14 tumors imaged from that microarray. 42, sorry. Additionally, we had pre-treatment samples from a number of patients who subsequently received immune checkpoint inhibitor therapy. They were treated with either anti-PD-1, anti-CTLA-4, or a combination of the two. And we have clinical information for them. So 14 responded and 16 did not respond to this treatment. So here I have a few example images of the kind of data, raw data that we're getting out of the CytOF machine. Here I've really zoomed in so you can see what we're looking at, and I've chosen just 3 choice markers for each image so that I'm able to color them and we can visualize what's happening there. All 35 markers stacked on top, it's hard to see. So in the first image here, I'm showing histone H3, which is a nuclear marker, says where the cells are. CD68, which is a monocyte marker, and CD3, which marks lymphocytes. And these are lymphocytes and monocytes are 2 very different types of immune cells, and you can see the expression pattern doesn't overlap. In the next image, I'm showing CD4, CD8, and FOXP3. These are all different types of immune cells, and the expected binding pattern here is that CD4 and CD8 should not co-occur. These are 2 different types of cells. And FOXP3 should always occur with CD4. This is a subtype of CD4 positive cells. And again, we can see this nice binding pattern here with little overlap between the green and the blue and a lot of overlap between the red and the blue. Finally, I have an example image with S100, SOX10, and CD3, the first 2 of which are cancer markers, so they mark melanoma. And the third is a lymphocyte marker. So again, 2 very different types of cells. They don't overlap between CD3 and these other 2, but we have a lot of co-occurrence for S100 and SOX10, which should both be marking melanoma cells. But what to do with this, with these nice images that we've generated? Here's an overview of the analysis pipeline that we've applied to this data. The first problem, and honestly a very difficult one, is cell segmentation. Where exactly are the cells? How do we define them in the images? The next step was cell type clustering. So once we've defined where the cells are, we use other markers to determine what the cell type is. Are they cancer? Are they T cells, etc.? Then we perform dimensionality reduction to better visualize all of these markers. How are they related together? We were able to perform cell type content analysis. We did a lot of different comparisons looking at cell counts, cell type proportions. And finally, spatial analysis, the big benefit to doing imaging with this type of technology. So the first step, cell segmentation, the cells are defined using the nuclear marker, in this case, histone. And here on the left, I'm showing a zoom in of the raw data that we have for histone markers. This we input into a machine learning program called Elastic, where we can perform manual annotation of the pixels, where a human says where the pixel is definitely positive for a nuclear marker or definitely negative. And after doing a number of annotations, the program learns from this input and is able to generate probabilities. There's a single probability per value image on the likelihood that that pixel is positive for a nuclear marker. We then input these probability maps into Cell Profiler, which then which first identifies where the nucleus is, and then adds a border around the nucleus, expanding to the cytoplasm and the rest of the cell to be able to pick up more cell signal. And then we've defined these shapes here as the cells in our study. The next step is cell type identification. So now that we've identified where in the images the cells are, we want to say what types of cells they are. In this particular antibody panel, we had 12 markers that were used to identify cell types, and they're defined here. The marker CD20 is used to define B cells. CD31 defines endothelial cells. We have a combined cluster for CD68 and CD14, which are macrophages and monocytes, because these are very closely related cells, and in our data, we found it difficult to separate them out. So they're identified as a single cell cluster. Additionally, we have CD4 positive T cells and CD8 positive T cells. Now, for these two types, we did further sub-identification using FOXP3 and CD45RO. FOXP3 is CD4 positive T cells that have FOXP3 are considered T regulatory cells, and then CD45RO, if it's present, are considered antigen experienced cells or naive cells. Now, the way we did this clustering was for this first step that is boxed here, we used the clustering algorithm called phenograph in combination with k-means clustering. So what was done was we did unsupervised phenograph clustering on all the markers in this box. And from there, we selected seed clusters that had expression with known biological significance. So any clusters that had high expression of CD20 and low expression of everything else, that was defined as a B cell cluster. We then use k-means clustering to collapse down all the other clusters into these biologically significant clusters. For the sub-classification, we again use the Elastic program that I described above. However, rather than doing pixel level probabilities, we did object level probabilities. So we trained the model on segmented cells, and we looked at cells and we were able to say whether they were definitely positive or definitely negative for FOXP3 and CD45RO. And then we were able to apply it to the already clustered data, where we defined positive as any cell that has a probability 0.5 or greater of being positive for that, and then lower was just determined as negative. On the right here, you can see a summary heat map of what our results were from this clustering method. I'll pull out a few examples. So CD31 is one that looks nice. You can see it has very high expression of the CD31 marker here, and low expression of the rest. B cells as well should have high expression of CD20, low expression of the rest. So as you can see, there is some noise sometimes, and there's also a lot of variation in intensity, since what we're looking at here is isotope counts. But what was really important was that the value that we used to call that particular cluster is high. Oops, sorry, is higher than the other markers for that particular cluster. Using this method, we were able to segment and classify over 220,000 cells. And as you might expect, looking at tumor samples, the majority of the cells are, in fact, tumor, melanomas. The next most common is others. And these are likely cells that we're just simply not able to identify, because even using as many markers as we did, they're eligible, that we can't cover them. Our suspicion, without having data, is these are likely keratinocytes, other cells that are typical to the skin that we just didn't have markers to be able to tell what they were. The next most common cell type was macrophages and monocytes. There are quite a lot of these, with the other immune cell types having much lower total counts. So here you can see how we've gone from the raw data shown here on the left. And what I'm showing here is the SOX10 marker, which marks melanoma cells, CD1468, which mark macrophages and monocytes, CD3, which marks lymphocytes, and CD31, which marks endothelial cells. And on the right, we have this cartoon where we've drawn where the cells are, how we've defined them, and then colored them by the cell types that they are based on our clustering method that I described before. And you can see the correlation looks pretty good. But what we're looking at here when we find these cell clusters is a rather large matrix of data, 12 different markers for over 220,000 cells. We're interested to collapse that down and see the relationship between all these markers. And to that end, we used the UMAP dimensionality reduction. And here you can see some nice clustering. The melanoma cells tend to cluster together, although there's a second cluster up here. I'll talk about in a moment why this might be. The most different type would be endothelial cells, CD31 positive cells. These are they're not like melanomas, they're not like the rest of the immune cells, and they tend to cluster quite far. Then we have this arm over here where it's all the other immune cells. First, the macrophages here on the left. You see a lot of overlap between antigen-experienced T cells and naive T cells. These are very closely related. Of the 4 markers that define them, only one is different between the two. Same for antigen-experienced T helper cells and naive. There's a lot of overlap there. And then you have this little arm coming off the top for Tregs, which has in common with the T helper cells CD4 expression, but additionally FOXP3. And then on the end you have B cells, which are a bit different from the rest, and you see them clustering a bit farther. What's fun with these plots is in the previous slide, I've colored them by the clusters that we've defined, but we can overlay all sorts of different data on this UMAP plot. So here we're looking at the intensity data. So for each cell, what is the actual expression of all the different markers that we used in our panel? So as I mentioned in the previous slide, you see there were sort of 2 clusters of melanoma cells, one in this lower left area, and then another one up here. And you can see when we look at the expression of our two melanoma markers, SOX10 and S100, the lower cluster expresses high levels of both, but this additional one expresses SOX10 and not very much S100. So this is probably just some nuance in small differences between melanoma types. And a good reason why we included two different markers to try and pick up as many of these cells as possible. Additionally, you see in that arm I described earlier, endothelial cells have high expression of CD31 over here. The T regulatory cells that I mentioned have high expression of FOXP3. And so we can see that the expression level matches up well with the clusters we've defined. And so given our visual verification of the images, as well as the way the clustering came out in the UMAP, we feel pretty good about our cell type identification. So now I'll get into a bit of the different analyses that we did now that we have all of these cells defined. So one of the first things we did was simply to look at cell counts. What cells are there and in how many numbers? What I'm showing here is every column is a sample, all 72 samples in our data set. And then every row are different types of immune cells that we looked at. So interestingly, we see that macrophages and monocytes, which is the most common cell type in terms of numbers, occurs in most samples. And then the next most common is T cells. And what we found interesting is that far fewer samples had T cells, but all the ones that have T cells also have macrophages and monocytes. And the same holds true for T helper cells with T cells. If they have T helper cells, they tend to also have T cells and macrophages and monocytes. T regs are even more rare, and then very few samples have any B cells. But the majority of samples that do have B cells also have T regs, also have T helper cells, T cells. And this was a very interesting pattern that we saw because it's been reported before in breast cancer. And so perhaps it's something intrinsic to the way in which the immune system invades the tumor microenvironment. So in the previous slide, I was looking at just raw cell counts, like if, are there, so here, sorry, I forgot to mention, we define multiple thresholds. If the sample has at least 30 cells, we've colored it as light blue, at least 50 is yellow, at least 70 is in red. And we call this cell presence, and we define presence at different thresholds. We wanted to see if this relationship holds when we change that threshold. But these are really just counts. And some samples have many more cells than others. So we're also interested in these samples that have a lot of cells or few. What are the proportions of these cells? Is there something to see there? And so here is just sort of a general overview of all the proportions, again, for all of the samples in our study. And some just general observations that we were able to see looking at this summary is, for example, B cells, which are quite rare. There's not that many, and not very many samples have them. There's two lymph node metastases with B cells. Additionally, we saw brain metastases tend to have fewer immune cells, but it's tricky to do statistics because we're working with relatively few samples in this case. But there are a number of other analyses that we can do, notably looking at the immunotherapy response, because as I mentioned, we have one data set of samples that are pre-treatment samples from patients who later received immunotherapy. So one thing we wanted to do was look at, is there some feature in the tumor microenvironment that correlated with these patients' later response or lack of response to this drug therapy? And looking at T cell proportions, we found that there was a trend for patients who responded well to immunotherapy to have higher levels of antigen-experienced T cells. However, it's not significant. But if we look at a subset of these cells, so Ki67 positive immune cells, and what Ki67 indicates is the proliferative status of these cells. So if the cells are positive for Ki67, they tend to be dividing. They're active. And we found for proliferating antigen cells, there are significantly higher proportions in patients who responded well to immunotherapy. So this was an interesting finding, and it was nice to see that this also holds when we look at survival. And the patients who have high proportions of proliferating antigen-experienced T cells have significantly longer survival times than the others. And here on the right is an example image from a patient who did respond to immunotherapy. And in green, you can see we've colored these cells using CD8, CD4, Ki67, which are the important markers in defining this particular subtype. And then in red is cancer. And you can see there's a lot. It's very well mixed into the tumor. So now we've talked a bit about cell counts. We've looked at cell type proportions, but we still haven't looked yet at the spatial relationship, the big important point using imaging data. So there's a lot of different questions you can ask about spatial relationships depending on how you look at the data. So methods in order to characterize the spatial relationship. The first of which was Delaunay triangulation, which I'll explain in more detail, mean and median distance between cell types, between cells that are near each other, and between for example, tumors and other nearby cell types. We looked at assortativity, which is the amount of mixing that we're observing in the tumor. And we also performed permutation of cell labels surrounding the melanoma in order to evaluate enrichment of contacts. So the first point that I'll talk about is Delaunay triangulation. So this is a pretty general method that can be applied to any set of points on a plane. And what we've done is we've defined the center of every cell as a point on the plane. And we want to find a subdivision of these points into triangles so that no point lies inside the circumcircle of any triangle. So that's what's shown in this part of the plot here. And the edges of these triangles have the property to connect adjacent cells from the corresponding Voronoi diagram, which is what's shown here. And basically what this means is we can draw connections between cell types that doesn't allow crossing of edges, because this would imply that we're not actually connecting the cell to its most likely contact. It also maximizes the minimum angle of all the angles in the triangle. So again, basically what this means is we're drawing contact edges between cells that are most likely to be in contact. And if we apply this to our data, this is the kind of spatial graph that we get. On the right here, I'm showing the Delaunay triangulation. It's overlaid on top of the raw data. So the blue is the raw nuclear signal. And then we have the point in the middle, the central point of our defined cell. In red is the Voronoi diagram. And then in black is the Delaunay triangulation, which connects these cells to say these are its neighbors. So for example, this cell here has 1, 2, 3, 4, 5, 6, 7 neighboring cells that we say are in contact with it. We found that cells typically had 5 or 6 neighbors, And the mean distance to the next neighbor was 16 micrometers. However, this mean distance between them varied a lot depending on the cell type. So for example, at the top here, we have lymphocytes, which had a median distance from a lymphocyte to its neighbor of 13 micrometers. And then this was the smallest distance. And then the farthest was between endothelial cells, CD31, and melanoma, which had a median distance of 18 micrometers. So again, I'll go back to this example spatial graph, because another interesting thing that it allows us to do is draw interconnected regions. So cells of the same type, we're able to make islands of them, essentially. So, and we've shown that here by coloring the edges between points of the same cell type. So for example, here you have a bunch of, a bunch of melanoma cells that are all together. Here you have in blue the macro mono cluster, and what we found here was the majority of interconnected regions, more than 50%, are generally composed of only one cell. But there's this long tail distribution with more than 50% found in melanoma cells. So, not 50%. With melanoma cells having the largest regions, followed by B cells and others. Sorry. And over here we see that the region sizes for lymphocytes tends to be smaller, and then you have very large regions of melanoma cells and B cells that are grouped together. So this tells us about areas that are interconnected, but what we're also interested in is the organization of these areas. And in order to measure this, we use the assortativity coefficient, which basically tells us the tendency of objects to associate with similar objects. So, over here, do cells typically tend to touch cells of the same type, which would mean they're assortative, have a positive assortativity coefficient? Is it random? Cells touch any type of cell, a coefficient of 0? Or do cells tend to touch only other cell types, which would be disassortative, a negative value? So this was a method published in Physical Review in 2013 and can be found in the iGraph package in R. So this gives the likelihood of cell contacts. So as I showed before, a few slides earlier, we have a lot of very large regions composed of melanoma cells and B cells, and macrophages and monocytes. And we see this corresponds well to assortativity. They tend to have higher assortativity coefficients, which means they tend to touch cells of the same type more. So we see the highest coefficient for melanoma, others, macromonos here. But interestingly, we see a very high coefficient for CD31, even though CD31 is generally makes up relatively small interconnected regions. And this is something that you can see even in the raw data. When we look over here, CD31 is purple. These mark vasculature. So even though the vasculature is relatively small compared to, for example, the tumor area, it makes a very defined structure. Like they really tend to clump together, and you can see that here in these ovoid structures. So this really highlights the importance of looking at these types of relationships in many different ways, looking at the Delaunay triangulations, distance between cells, and these different kind of calculations that we can do. Here are a few more examples showing different assortativity scores. So in the top row here, we have vasculature, CD31. The one that I mentioned was highly organized, and we see these higher assortativity values where you can clearly see the cells tend to cluster together here and here. Slightly lower value here where the vasculature is a bit more spread. We also saw a huge variation in the tumor structure. So melanoma assortativity varied quite a lot from 0.1 over here, where you can see there's a lot of infiltration and the tumor is fairly spread, to over on this side where the tumor cells are pretty packed and there's not a lot of other cell types infiltrating that tumor area. And you can see this in the assortativity score given. So another method that we used to evaluate physical relationships in our data set was to simply look at the distance between melanoma cells with every other cell type present. So we're really interested in the relationship between the tumor and everything else. And the most significant one we found was that melanomas tend to be quite close to closer to antigen experienced T cells in patients who responded well to immunotherapy. So this was very interesting. This was, you might remember, this is the cell type I mentioned before, where having a higher proportion of these cells was also indicative of response to immunotherapy. So it seems that it's not just the quantity of cells there, but also its spatial relationship with the tumor that's important in determining response. However, just looking at this distance approach, it can be confounded by cell type proportion. So another method that we applied to our data was the permutation approach. And this is where we kept the melanoma, the tumor cells, stable, and we shuffled the labels of every other cell type around the tumor in order to establish a null distribution. Allowed us to measure contact enrichment between melanomas and the other cell types. And we found high contact enrichment between melanomas and the macrophages and monocytes here, and very low contact enrichment between melanomas and CD31. And when we looked back at these antigen-experienced T cells, we see that they are in fact enriched to be in contact with melanoma cells. Not quite significant, but the trend. But we do see a trend that is consistent with the distance approach that I presented previously. So given all this information, what we're seeing here is that it's the proportion of proliferating antigen-experienced T cells in the melanoma microenvironment that is important in determining response, and additionally the distance between these antigen-experienced T cells and the melanoma. The nearer they are, the better the response seems to be in these patients. And so with that, I'd like to thank everyone else in the lab who has helped work on this project, particularly Mathieu Lajoie, the other bioinformatician, and Dan Moldoviano, who generated all these images and did all the antibody work. And thanks to Ian Watson, our supervisor, for supporting us through all of this, and of course the various funding agencies that have helped pay for all this data for us.
Great presentation. Thank you so much, Ms. Ramsay. So going to the question. Someone have a question? All right, so I'll start the question. It's about the spatial graph that you are using it. Which tool you used to check the edges and the
Which tool?
yeah.
So this these plots were all generated in R, and you can do. Let me just double check that it's I believe it's the iGraph pack. Oh no, sorry. It's the DLDIR DLDIR package that can perform Delaunay triangulation and generate these kind of graphs.
Perfect. And about the counting of the cells, you said that you use the threshold. Is it depends on the literature, or you just use random threshold?
Yeah. So what started us down that road was there have been previous studies that presented if you define the presence or absence of cells based on a threshold, that they saw this particular pattern of a lot of samples have macrophages and then T cells, et cetera. And so we applied the same method and found that in melanomas, we saw the same pattern, which was interesting since the other paper was on breast cancer.
Nice. And for the last question for my questions is elastic package that you used. Do you train your own model, or there is a model that is already trained and you just take?
Yes, we trained our own model. So for that part of the analysis, it's quite time consuming, just going through one image at a time and labeling what's what. But it works reasonably well, so it's worth it.
How long it takes?
Oh boy. I mean, each image maybe less than 5 minutes, but if you want to look at every single image, 72, it adds up.
Can't imagine. There are no questions on the YouTube channel. I don't know if you have questions. No? Perfect. So thank you so much, Ms. Ramsay. So if you can go to the link Wonder for a networking session, and yeah, see you there. Thank you so much.
Thank you.
Thank you.